we have created two classification notebooks, one for document classification and the other one is the DNA sequence classification. So let's go through some steps. I think most of the components of these two notebooks you are already aware of. Let's go briefly, starting with the document classification. Okay, so here I'm loading my packages and then I put a data description. So we'll be doing a document classification for 20 news groups data set. Okay. And this is under the sklearn dataset package. So here I'm loading the dataset and I'm actually getting four types of documents. One is religious documents and computer graphic, and then science medicine, okay, so atheism. And then let's look at the data side. So we have about 2,500 documents. And you can see like here we are using the count vectorizer, and ngram one means that we are using only the unigram as features for the count vectorizer. So now we have each document being transformed into 35,000 unigram tokens, So if you pass 2 by 2 tuple as parameter, then keep it in mind that we're using 35,000 unigrams. So let's look at how the feature vector size is actually changing. So you see 35,000 now become 264,000 features, So we have both unigram and bigram features, So bigram not meaning just only bigram. Bigram meanings both unigram and bigram. Just keep it in mind. So I'll do experiments with just using the unigrams. So I redo my run. And now we are assigning the X trend counts as my X and Y to do the modeling, but you can always look at the detail of what the feature values are, okay? Now I'm just looking at that what is the highest frequency token, and it found to be the highest occurring token. And you can actually print the vocabulary. You can look at what are the elements in the count vectorizer, and so each token or all they have an ID. Feel free to go through all the parts of the notebook and just let me know if you had any question. So now I'm looking at what are the different class types we have, and if you recall we loaded 4 different types of documents. So essentially we have 4 class and these are the class labels. So I'm doing an experiment for 80-20% XSplit and using logistic regression as my classifier. But essentially you can run any classification methods once we have pre-processed our features and labels, okay? So now the training is done. Now we'll be looking at the accuracy, and you can see that for 80-20% setup, we have 96% accuracy by the logistic regressor classifier. And here we're looking at the confusion matrix, and these parts you are already aware of that because we are reusing the same notebook but just passing new data to do the classification task. Feel free to run all the remaining part of the notebook to see how the other models are actually performing, but we can see that the logistic regression classifier is really doing good for class 0 and class 2. But as we always say that we have space for improvement, no worries, okay? Now let's do the same for the DNA sequence classification. And here again, I have put the description of the dataset. So here we are using one dataset from Kaggle DNA sequencing. And I have provided the link so you can go through and look at the details of the data. I have also put the data in my GitHub repo, so you don't need to actually download the data, rather you can just load the data by using these links. So here I'm loading the data, and actually they have genome data for 3 different animals, chimpanzee, dogs, and also for humans, okay? And here we are looking at some portion of the human genome data, and this is the function that we have discussed in our presentation part, KMARS. So this will actually convert these sequences into sequence of tokens or words, whatever you call. And here I'm actually showing the same instance of the KMAR formatting that we have seen in the presentation, so you can feel free to run and check. And so I'm doing it for all the data, human data, simple data, and dog data. And after the KMR encoding or formatting, reversing, whatever we call, you see that now we have all the documents being labeled as class and all the tokens, and again these are kind of a document by splitting the words, and words are actually the overlapping substrings that we have explained in our regular presentation. Okay. Here we are looking at the distribution of different classes. So we have 7 classes of human DNA in our mix, and we can see that the class 6 is actually have the most number of instances, around 1400, and the class 5 has the least, about 200 instances. Okay. So here we are doing the count vectorizer by passing all the documents or DNA sequences formatted into document, and I'm fitting my count vectorizer, and here only using the unigram, just as we have done for the document classification. Okay. Now the fitting is done, and I am transforming all the features in. I will print here the shape of the matrix, and so you have about 4,000 records, and number of unigram feature is about 4,500. Okay. But again, you can extend it to bigram, and all the feature encoding are done by count vectorizer, as we have done for the document classification experiment. Okay. Now let's look at the class level. So you can see that we have 7 classes, as you have seen. We are doing the same 80-20% training split, and we are fitting a logistic regression model. Okay. So here we are just learning a classifier for the human genome classification, okay, by using count vectorizer as our features. So we have done our model with our model fitting, and the accuracy is 91%. Just like the document classification, we are doing a confusion matrix visualization, and we can see that class 2 is perfectly classified, almost around 100% accuracy. Class 5 doesn't look that bad, but the class 3 and class 6, they are actually struggling to get better accuracy. Okay, and also class 4. Okay. So here again, I'll share these notebooks, so feel free to actually run through these notebooks and try to get more understanding of the videos or experiments, and feel free to reach out to me if you have any question. Okay, thank you.